Everybody, hi. On this International Women's Day, Element debuts a series of fireside chats that focus on women's leadership. For our first episode, I'm really excited to have 4 wonderful, talented women scientists and leaders in genomics joining me. We will learn more about their work and science, hear the views of where the field is going, and share their personal journey as women leaders in science. And I'm going to introduce the panelists now in no particular order. Catherine Aquino, Head of Genomics Analytics, Functional Genomics Center, Zurich. Eleanor Carson, Associate Professor, Bioinformatics, Integrative Biology, UMass Chan Medical School. And she's also a Director of Vertebrate Genomics at the Broad Institute. Anoja Pereira, Director of Sequencing and Discovery Genomics at the Stowers Institute. And last but not least, Jasmine Plummer, Director, Center for Spatial Omics, and she's also Associate Professor, Department of Development Neurobiology at St. Jude Children's Research Hospital. Thank you so much for coming today.
It's a pleasure to be here.
Yeah, really, really happy to have you here. I know of your work to some extent, but I would love for you to kind of share with the entire audience, what is the focus of your current work? And if you can name just one thing that truly excites you, what that is. So who wants to start?
So at Stowers Institute, we focus on basic research. And I think that's pretty exciting because that's very unique. And I get to work with all sorts of model organisms that are standard as well as non-standard. So I feel like every time we get a project, it's something new that we have to sort of optimize and get going. And we have labs that studying regeneration, diabetes, and for all these different things, we are using very novel organisms.
That's awesome.
So
yes.
I'm at St. Jude, but I just recently moved here. So I have a long background in genetics and really trying to not just move basic science into translational, but working very well with translational into clinical. So a lot of my allies have always been clinical geneticists who actually diagnose tumors. And really what I'm most excited about now is kind of using all these genetic tools, and I myself am a PI in the Human Cell Atlas, so taking single cell tools, all the sequencing tools we have, and really bringing it into the spatial realm of being able to delve into tissue and patient samples.
Awesome, great. Thanks, Jasmine.
Yeah, and I can go next. So my work is focused on, like many people on this meeting, figuring out how to understand what DNA does. You know, if we can look not just at the DNA of humans, but of many different species, can that actually understand how a particular change in the DNA actually ends up changing, you know, the animal or the plant at the end of the day? Because, you know, DNA is just this long strings of A's, G's, and T's. So it's kind of this amazing leap that happens somehow between those two pieces. And I think what I'm most excited about going forward is the technology has, you know, come so far over the last few years that we're finally at a point where we really can start thinking about how do we not just find things in the genome that are correlated with, you know, for an example from my own work, I do a lot of work on dogs that are correlated with whether a dog is interested in retrieving a ball, but actually figure out how that change is changing how the dog's brain develops and how that changes their behavior, and not just by having to like look at the animals, but now we can do it in cells and we can do it in tissues and we can do it in the lab and really start picking apart how things work.
Yeah, that's pretty amazing. I actually was in your AGBT talk. It's fascinating how this dog genetics is so interesting because, you know, the breed and the phenotype is actually doesn't necessarily correlate all the time. So Catherine.
Yeah, my talks these days tend to be a bit of a series of figuring out how many puppy videos you can put in a talk and still maintain some kind of scientific credibility. Yeah, I don't think I've maxed it out yet.
There never is a max. Everybody loves puppies. You chose the right model organism.
Catherine.
I guess it's my turn. So yeah, so I work at the Functional Genomics Center in Zurich, Switzerland. And so we are a core facility of two universities, which is the ETH in Zurich and University of Zurich. So basically, even today, according to my LinkedIn, because I just updated it, I've been working at the center for 19.3 years.
Wow.
And so,
Right! That's impossible! You look too young. We need to study those genetics.
That's true. We need to sequence Catherine.
Yeah. Yeah, and the thing is, so basically, I really like this line of work, obviously, because I've been there for a while, and I've been excited for 19 years actually, because we started with microarrays, and then now with the sequencing, we went through the whole thing, 454 solid, the whole thing. So I've been excited for a lot of things. So every year I'm very excited about something new, all the new toys. Yeah.
So that's the feature of scientists, right? I remember realizing once that we were talking to a group of non-scientists and they're all like, so what did you figure out with your research? And all the scientists were like, oh, we figured out this thing at the end of the day. But actually what's really interesting is whatever the next question is. Like once we've solved the problem, we're always on to the next thing, the next new exciting unknown.
Yeah, we don't have our own biology, but then since we support a lot of different biologies, and always get excited about a lot of the different research projects and to use the new technologies to answer diverse research questions, I find that they're very exciting. So I'm excited every day.
I can address what I'm excited about, and I would say that I started as a technician working on the human and mouse genome projects, and that was over 2 decades ago. And we were doing slab gels. So I have seen the progress that sequencing has made over the years. And I'm really, really excited right now to be in this single cell spatial space and also see all these new sequences coming into the market.
Exactly.
Yeah, that's really interesting because I will raise my hand because I did slab gels every day in my graduate school to make point mutations, out of one protein. Really just amazing to see how the technology has progressed. And all of you touched on, interesting toys and tools to use, in the genomic research. Because you're obviously in kind of studying different subjects, right? Because Anoja and Catherine, you're more of like core lab. You have all kinds of different toys. Jasmine, you're in the more of a translational space, wanting to get a clinical, and Eleanor, you're in the more of a plant and animal space. But there is a commonality here. I think that really is how these innovative tools enabled your work and enabling the future discoveries. So my question for you is kind of maybe zoom out a little bit. We talked about your own work, zoom out a little bit to look at the entire genomics field. What is the most exciting kind of breakthrough, whether it's tools or some discoveries, scientific discoveries that you personally are very excited about? Just wanted to open up the floor and see what is the most exciting thing for you in genomics research today?
I can start. So I actually am an example of everybody on this call because I started in C. elegans. So I was very lucky in my training. It's a small model organism and I got to do whole genome sequencing. And that, made the leap now only into human and mouse, primarily servicing mouse labs. That's not to say we don't have other model organisms that are super important, but I think that's why everybody's on this call. It's not about that we just study human stuff. And having come from C. elegans and going into human genetics, it's always been like exactly what Eleanor had talked about before, is like, what is that mutation, right? Because fundamentally we can look at RNA, we can look at protein, you can look at metabolomics, you can look at whatever you want, but really there is hopefully a fundamental change that we're all chasing in the DNA. And that excites me the most because the field has really evolved to incorporate every organism known to man, but also really translating like simple things that I think we all knew and we were all chasing, but is kind of real now, where like, we know there were these genetic perturbations, right? And we would screen for BRCA1, But from BRCA1, now, if you have a BRCA1 mutation, it might set you up for cancer, which would really suck. But now you have these immunotherapies that we now put your tumor on, and we see like we can targeted therapy now. And really everybody on this call, while you're talking about model organisms, it really stemmed from that. And I think that is what really, I think the technology is telling us.
I think we were just really talking about how this field has evolved, in terms of tools and also scientific discoveries. From, in the tools part, we started from slab gels in many iterations, and now we're seeing a bloom of sequencing technologies today in the market. And also we talked about how these tools enabled, all of your research. So, Jasmine, you were talking about what is the most exciting thing for you in current genomics field?
I think for the first time, we're really seeing a clear path to what everybody, I think, on this call is. We all have our interested phenotypes, but we hope that at some point they make a change in human disease as far removed as plants or animals or model organisms. And I think we had that dream in genetics a long time ago of putting things on a sequencer and altering kind of treatments, but we're seeing that now. So, I gave an example, I think, of BRCA1 mutations where we knew to screen for that. But now, not only can we screen for that, we can take a woman's tumor and say they'll do better for immunotherapy. So I think we're not just talking about plants and cool Drosophila or C. elegans or model organisms. We're actually, in a way, using that combined basic research and science. It's not this removed system. And I think COVID showed us that too, is that we're not as far removed from basic science into true applications. So that's the thing I'm most excited about, especially as we move into spatial, because spatial really is, I think everybody wants to see it, right? If you have something that you think is a mutation, don't you actually want to see how it perturbs something in the tissue?
Yeah, that's really great, because I do want to kind of write that train of thought, because you mentioned that, you're now seeing the transition from fundamental research to clinical and translational. What do you see as the biggest roadblock for that to happen? Or, in another word, what should happen, what has to happen to promote these kind of quick acceleration from research to clinical space?
I mean, I think one of the biggest challenges we have right now is that our way of coming up with new drugs and testing new drugs is not particularly effective, in the sense that we have many more ideas at the beginning of the day. And by the time you get into clinical trials, and then into an actual drug, it's a very small percentage of it actually ends up producing something that's effective in humans. And it's often not super effective. It's often, kind of an incremental change, or you need to worry about side effects. And one of the things I'm really interested in is that now we have new technologies that are giving us new opportunity to kind of treat any species as a model in a way, because rather than having to actually have it in a laboratory mouse kind of model where you can control everything, which is going to always be valuable, but is a very artificial system, can we actually start looking at animals living in the real world, and whether that could actually help us discover new therapeutics that could be helpful. So one of the things that I'm really fascinated by is basically weird traits that have evolved in other species. And so things like hibernation, where you've got squirrels that can lower their body temperature to like 4 degrees Celsius for months on end, and they get, a bear will get super obese and then go and hibernate and lose all that weight really quickly and not move for months on end, and they don't have strokes, and they don't get heart disease, and they don't get dementia, and they don't get diabetes. And it's like, how are they doing that?
I'd love to hibernate. Can we implement that model?
Exactly. And so it's like, can we actually figure out how they're doing it? And rather than, trying to like figure out how to treat the disease, figure out how to make things healthier in a way as an entry point to drug development.
So at Stowers Institute, we work on basic research. We are trying to understand the mechanisms underlying human diseases. And to do that, we use both standard model organisms and non-standard ones. And to go to Eleanor's example, like right now we are working on this fish from caves in Mexico they're albino, they eat when there's a flood, they get really fat, and then they lose that body fat over time. But they don't have any sort of like diabetes in them. So there's a lab that's studying that mechanism. To go back to the question of, what needs to improve going forward, I have been I started my career as a technician doing slab gels over 2 decades ago for the human and mouse genome projects. And a problem that I have seen that has sort of improved, but not really, is bioinformatics. The amount of data that we are generating keeps increasing, and it seems like we are having a very tough time keeping up with all the data, the storage, the analysis, all those aspects, and that has to improve.
People ask me what I'm most excited about and also what I'm most worried about. It's basically the same thing, which is that I'm really excited that we're going to be able to sequence everything in existence. And also I have absolutely no idea what on earth I'm going to do with all of that data and how I'm going to analyze it and store it and make any kind of use of it.
Yeah, that's really great.
I'm also concerned about democratization, right? And so we are very privileged. The people on this call are very privileged in our access to the types of tools and instruments we get to use. And I'm very fortunate to be part of studies that are not part of H3 Africa, which is amazing and very well funded, but really like true grassroots kind of organizations that want to have access to kind of all these amazing discoveries that we're making, but really, how do you put a -80 in Africa, right?
Yeah.
How do you actually put these equipment in when there's no emergency outlet, right? I was on a call recently about best practices for like how to preserve these things. And the surgeon on the call was like, room temperature is the doors open, right? Like it's not, the windows are open. Room temperature is 43 degrees Celsius sometimes, right? And so I think that we talk about all these things, but we really just own them in certain spaces. And I think especially when we talk about genomics and why we're on this call, it really is the people that are probably going to be most useful for this don't have access. And how do we do that? And that's one thing, again, with the spatial. One thing I've learned, and I didn't learn this when I got into spatial, I'm learning this after for the democratization, is like everybody can put things in FFP. They might not do it really well, but they can do it in underprivileged countries. They can put it, that's how they diagnose tumors still, or how they diagnose IBS or Crohn's disease. And so I think in that sense, being able to really put things in a place that they can at least ship it to us or do it that way. And then back to the bioinformatics, that's also a way I think we can democratize things, is that it's not so difficult, once you get internet established in those things, to allow them to have access to their data and access to their data and really teach them, that's a larger problem, but teach them how to actually do the bioinformatics in-house.
And Jasmine hits on a very important topic. And I would add to that by saying, when we do human genome sequencing, we are not capturing all the populations right now. And for us to really improve human health, we need to do that.
I want to add something. So I mean, Africa is far, and I'm also coming from Philippines. So, but actually, within our within the university in Switzerland, even so, not everyone still has access to these technologies, and I find it's my duty as a core facility person to give access to provide all this, set up the new technologies, implement it, and give everyone access to it, and then we can disseminate this this knowledge to other places, for example. So we have different we have projects in collaboration with Indian partners, and so we try to do that in collab with the Swiss Next. Actually, it's a Swiss network for innovation in science. So we try to do that as well.
Yeah, that's great. Those are really all great points, because as the sequencing technology develop, the spatial and all that develop, as we can sequence more and more people and species, the data is becoming a bottleneck. And also, to many of your point that democratization of sequencing and all these tools extremely important. This is actually happens to be Element's mantra, right? So Element's Biosciences really is trying to increase access of sequence to everybody. So that resonates very well with me. Now, I wanted to kind of pivot a little bit to the topic is, really the women's leadership, in particularly women's leadership in STEM, in science and technology, because you are very accomplished women scientists and leaders. You've done so much great work in the past, many publications. What is your biggest challenge you have ever met as a woman, when you were, maybe backtrack 20 years ago or 10 years ago? What is the biggest challenge you have met, and how did you overcome that?
How much time do we have?
You can have all day.
I wouldn't say 20 years ago. How about today and yesterday and the day before that?
Yeah. Do you actually see a progression? No, that's really great thought, right? So what was your challenge 20 years ago, 10 years ago, yesterday, and today?
I mean, if I'm honest about kind of even our training, is when I started, I felt like there was fairly equal representation. It was still swayed, but we all entered kind of graduate school, and very few of those people that I entered graduate school with as females have stayed in science. And that rings true today still too. And so I think that while we can sway the STEM and get kids trained earlier and get them interested. I have 2 daughters myself, which speaks everything to like how difficult it's been as a female leader. But I would say that mass exodus of us at a certain point has, in my mind, not gotten better. And I think COVID made it worse. And I think that still rings true to me every day when I look at all the friends I had in graduate school or in my early or even earlier training that were smarter than me, had very much more things to offer. And I think there is a female viewpoint that we're missing in science. We look at science, no matter what, everybody has an individual viewpoint. And I think that's the most exciting part about science, is that we push each other to question things in a different way. That's the whole point of being a scientist. And if you're missing a large portion of the population, I think you've already skewed how we're interpreting things.
So why do you think it's the case? What do you think is the reason behind this mass exodus of women in science as they kind of move along their career?
Childcare.
Well, I actually think it's a question that needs to be asked better than it has been. I think that society's biases come through even in the answers that we just automatically give to questions like that. I remember a few years ago, there was an article in the Boston Globe just looking at the size of startup packages that new faculty were getting at universities in the Boston area. If I remember correctly, I can't remember exactly what the numbers were, but the startup packages that women were getting as new faculty were dramatically smaller in terms of their monetary value than what was being offered to the guys. And this is not necessarily anyone going in and saying, we're going to give the women half as much. This is the challenge, right? Is that a lot of this stuff is not overt. It's just kind of part of the system, and you're not even aware it's there until somebody asks the question. And at that point in time, it was like, I started wondering, I was like, everybody kind of says, well, women are leaving for reason X, Y, and Z, which may be true, but how much of them then gets influenced by the fact that their science is that much harder to do because they're not being given the resources they need to hire the team that they need to support them in that work. And so I would be really interested in actually, and I'm not super familiar with the field, but actually kind of digging into that question a little bit more as to why it is that women are actually leaving, and what are they actually being given equal resources before they make that decision.
I would say also being at the table. And so, the further up you get, the more you don't see women at the table. And so it's not uncommon for me, and I'll be honest about this, to be the only female in the room for some of my meetings, many of my meetings. And I think that's just, I mean, I can't even imagine how discouraging that was. That was discouraging to me. And we're talking 20 years, right? We're not talking it's current days. And I think it's even hard in my space to find female mentors. And I value my male mentors. Absolutely. They've been guiding me a lot of the times, but also to Eleanor's point, it's also the perception, right? It's also the perception of how, you know, when she talks about that statistic, it's just one statistic, but I feel like there's this whole mesh of why we're leaving or why we stay or how successful we can be, right? There's still females in science that maybe not as successful in terms of grantmanship or those kind of things. I feel, and we're also being, there's a tone in the room too. And I don't think anybody articulates that, but I'm totally happy to say that. I've been called alpha female. Like, I don't know what that's supposed to mean, right? Or aggressive, or, and you know, there is kind of this mechanism that my younger self would have taken that in and taken it to heart. I think what's made me successful, which is really unfortunate, but that I have to adopt that, is just the thick skin and not let things affect me. And I think that's something everybody in this circle will have experienced for sure. And I'm not just saying that males don't experience as well. I would love to see kind of what Eleanor's suggesting is like, what is the true dimension of all of that, right? Because we pick and choose those points. I've just accepted it. It's unfortunate that I have, but I think it's a thing. I don't know. And then I don't think there's enough of us to talk that are talking about that actively in a helpful way, not a hurtful way.
I was very sorry, Catherine, go ahead.
I just wanted to say, you know, I'm coming from Philippines, and actually Philippines is very good at gender equality. So when you look at the list of gender equal countries, so I'm actually when I was growing up or in the university, I was very used to having more female professors than male professors, and then more female everything leaders than male leaders in the sciences. And then when I went to Switzerland, that's totally different, and that was actually very shocking for me. And I was thinking like, oh, so why is it like that? So somehow I don't have the answer, of course, but somehow it's natural. It's more more natural for Philippines to be having all these women leaders than the Western countries. And why, I don't know, but that's one thing that we're doing quite well in my home country.
I would agree with that. I would say I'm from Sri Lanka, an island in the Indian Ocean, and it's the same mentality there. The world's first prime minister, the female prime minister, was from Sri Lanka. And I was also very lucky to grow up in a family where my mother was sort of the matriarch, and she enabled us to do whatever we wanted to do. And we were always allowed to speak up. You know, if we didn't agree with something, we were allowed to speak up. And as Jasmine said earlier, it's important to be at those big tables, to speak up and be confident about it. And I think we all try to be good allies, right? Like even at one of the most recent conferences, Jasmine and I were talking about how we have to stick together. So making your connections, building your community are very important things, especially as women.
Although I will say that one thing that Jasmine said, I'm not totally sure that I agree with, in the sense that, Jasmine, you said, and not let it affect you. And I don't know whether that's ever actually true. That those
I know,
moments,
it's not true.
It totally affects you. You just have to absolutely do your best to not let on that it's bothering you, because, you know, that's, you know, the last thing you want to do in those situations. But it can get really tiring when you're kind of constantly having to deal with a situation where you feel like you're being perceived differently from the other people in the meeting based on not, you know, as a scientist, you always want it to be about your abilities, right? You always want to think that whether your grant got funded or didn't get funded was based on how well it was written, and not whose name was at the top of the page. But then you know, it's always kind of in the back of your mind.
Yeah, I had a colleague a few years ago tell me, because I was having a hard time. It's not easy because basically I'm very feminine and very small, and somehow that it unfortunately packaging matters. And yeah, and then unfortunately, I'm also very cheerful and I was very happy. And then yeah, so he said, "Oh, the problem with you is you're always you're smiling all the time. You're always happy."
I've heard that all the time.
Like it doesn't make me less competent just because I'm smiling all the time. So imagine,
someone told me once not to sing when I talk, that I have too much inflection in my voice.
Oh, geez.
I was like, I feel like that's how I'm made. I don't know how to do that. I have to go to voice therapy. I don't know. Anyway.
right. So if it was like a big white man who's smiling all the time, they will say, "Oh, he's so pleasant," But if it's like a little Asian woman smiling all the time, it's not that something is difficult, something is different. Something is wrong, and that's hard because if you so the message is, if you want to be successful, you have to kill something inside of yourself, right? And that's not fair. And I refuse to do that, so I'm still smiling.
That's
You want to
fascinating.
smile.
Yeah, that's fascinating. I can definitely relate to many of the points you ladies mentioned. And, as a matter of fact, I was the only woman in many, many meetings when I was in my past job for the entirety of 4-something years. I was the only woman director in the R&D for many, many years. And it's difficult, but I think, because we, especially me as myself, as Asian woman, I was taught to be humble, taught to be modest, taught not to, kind of cause a scene, those kind of things, right? But at the same time, if I don't speak up my mind, then we will be seen as incompetent. So it's really hard to balance these kind of traditional culture values that I have versus the need to really speak up to be a leader. And in the end, I realized, I cannot change myself. I wanted to remain humble. I wanted to be kind to everybody, but at the same time, I wanted to be a leader, and I'm not going to change who I am to be kind of better accepted. So I think, the authentic leadership really speaks very loudly to me. And I think eventually everybody will have, their own style of leadership. But I wanted to come back to all the challenges we faced, as women in science or women leaders in science. What do you think we can do together? Because I think, Anoja, you had a really great point, stick together, network with each other. Is there anything else we can do, or the society we can do, or we can promote the society to do to help more women in science or in leadership, to encourage that?
I'm going to comment briefly on what you just said, because I think the two sort of tie together. I don't think what we want to manage to achieve here is, we don't want women to have to change the way they're behaving to become, in quote, good leaders, because you're already a leader. And the old way of doing science, where you have a hierarchy and you have somebody in charge who knows best, or you have competition and everybody's keeping secrets and it's very cutthroat and competitive, that wasn't a system that necessarily led to the best science happening. And I think figuring out how to change science to actually be more collaborative and be friendlier and be more understanding that everybody's got their own perspective, and how do we actually move that forward, is kind of what we should be going for here, as opposed to trying to fit into the way things had been done in the past. I like doing science because I like working with a group of people that have a common goal. Like, I think that's fantastic, and I would hate to lose that.
So I'm going to pick up the thread of Eleanor's. I think I can't change institutional, or this is deep-seated from long time ago. But what I can do, and I think why we're all successful in our roles, is we're just still ourselves and being authentic to that process. And so part of my authenticity is that being a leader is that everybody beneath me that I train should be better than me, right? Like, I don't like if I've trained you well, or if I'm lucky enough that you're part of our team, then you become the specialist in whatever science that you are doing, and I will learn from you. And so I think that is like identifying the people in the room that are smarter than you and helping augment them. And I think that's true of even the circle. So going back to what Anoja had said, like we stick together, it wasn't an exclusivity thing. It was an uplifting thing, meaning the only way we're going to do this kind of not just at my team level, right? That speaks to me as a leader of a team, which I'm not. They all lead me and I get to show up and see the fun science. But also in terms of the larger networks, and the larger networks are like small things that, every time I can't make a situation or it's a conflict in my schedule, I do my very best to nominate another female or somebody in DEI. I absolutely do, and I know that seems swayed, and I'm putting it out there in the atmosphere only because their starting point is harder. And so, just to give value to, like, when I say no, hopefully somebody that I respect who has amazing science can be put in that same position.
I completely agree with everything that you just said. My team is mostly women, and that just happened by accident. Anytime I'm asked to give a talk, I always see who in my team is the expert in that, because I'm just the mouthpiece, right? I try to give opportunities for the people who are actually doing the work and have them shine. And I want them to be the future leaders that I know they are already. And yeah, I'm at this point, I'm not going to be like changing myself. Like I often hear I'm aggressive, I'm outspoken. I'm going to get worse as I get older.
It's about normalizing it. I think what is nice if people get used to the idea that there are women leaders and it's not unusual anymore, and then they will not think like I think we should also say, like, I should also say, like, sometimes there are women leaders but you don't notice them because they try to be as unidentifiable as possible,
Yeah.
So, don't look at me, I'm a woman. So I'm going to try to, how do you say, fit in. And so, yeah. So what I want to say is that we should normalize, being yourself in that people won't find it unusual anymore. Mm-hmm. And it will give, the new generation an easier time to enter the game if it's a game.
And then I would also say in that normalization is also the things that we get that are negatives, we actually, by normalizing, we highlight, right? So I will say one amazing thing is also being drawn. I've obviously made friends with these people outside of just this conversation because like attracts like. But really, one of the things I do see in kind of female conversations in science is like, we're very good at organizing and multitasking, right? That's just kind of how our brains, Eleanor will go figure that out for us, right? But that's the behavioral phenotyping, not for all women, but I think that's our strength, in our environment, especially in genomics, where the assays change every day. And I think like that normalization comes with that. It doesn't mean we have to fit in the other box. Because while our skill set may be a little bit different in terms of how we think, there is an augmented value in science for that way of thinking. And I think the more we normalize that and say, we can be spirited, we can talk, we can do all of those things, that also gives us value in science too.
What I would actually say is that every population has a diversity of phenotype, because I'm terrible at multitasking.
Oh, are you? Okay.
But I will also
you have
say
to be good at it, because you know every phenotype of a dog I've ever heard of. So
I know, I just have to keep
that is multitasking, actually. It's just, it's the categorization of how you think we're talking about multitasking.
There we go. No, I will say that I think, at the end of the day, I think the most effective research group is a research group that has a diversity of backgrounds and opinions and expertise in it, so that you avoid getting caught in that trap of everybody thinking about a problem the same way. Like you want your new graduate student to come in and actually ask that supposedly stupid question that actually isn't stupid at all and totally undermines what you've been doing for the last 6 months, but you're really glad they asked it now rather than after you wrote the paper. And that's, that's the problem with women in science and diversity in science, is that how do we actually start to shift the system? How do we actually make it so that more people are coming into science so that we can have that diversity of perspectives?
But don't you think sometimes that, before you get into a certain position where you can change things, you actually have to, toe the line and try to conform a lot. And then by the time you're in a position where you can actually change things, you're either just too tired or you have drowned in the system. Yeah. So yeah, because basically it's very difficult to get into a position where you can change things when if you have not if you're different from the beginning, right? Yeah,
I will admit to very often being quite tired. No, and I'm very, the reason that I got to where I got to was because of the people that mentored me along the way. Like that's how science happens. It's who you work with and what skills they can teach you to actually figure out how to make the system work for you. And so one of the things I get really excited about are things like research summer programs with high schools and things like that, where you're really getting the opportunity to get people when they're very junior in their career so they can start making those connections and coming in. And I always, it's always interesting when you look back on your career, you're like, well, if that one thing where I met that one person hadn't actually happened, I did a summer research program when I was an undergraduate at Baylor College of Medicine, which is kind of where everything built from after that. And it's a little scary how tenuous that is, but it's, the more we can actually support those programs and the people running them to actually keep those doors open for new people coming in, I think the more we can have that diverse research experience at the end of the day.
Yeah, that's really great point.
I was just telling my team right before this call, I hate doing this sort of thing, like be behind the camera. I hate it, but I feel like I have to put myself out there because I want to represent a population that's not normally represented. I want people to see, I want my girl to see a brown person on a screen and say, oh yeah, look at my mom, that's, she's doing that. I can totally do that in the future,
Yeah, thank you so much, Anuja, for your courage. Because I went through the same battle too, because I don't like to be behind camera myself, but I feel like I need to do this, have to do this, for the benefit of the younger generation. And I'm a huge fan of mentors and mentorship because I'm a benefit of a great mentors. I benefit so much from great mentors in the past. So now actually perfect segue to our last session. I just really wanted to ask you, you guys have tons of wisdom to share. What is your few words or a sentence of advice for younger generation of women in science? What will be your advice?
That's a tough one.
Yeah.
I think being quite stubborn is pretty important, to be honest. Like just being
I like it.
And not giving up easily.
That's truthful.
Yeah, I love it.
I would say passionate. No one in this room is still in science.
That's much more positive.
For whatever we've talked about today, unless you're passionate and you are your own champion. So lots of people, Catherine alluded to it already. And so is that there's lots of people that will knock you down along the way, but if you're passionate about it, you'll still want to do it because science is amazing. I still look at things all the time and just get really excited by it. And I think that's the best job in the world, right? Is that I get to discover new things every day and get paid for it. That's amazing, one. Yeah. And two is being your own champion so that every time somebody knocks you down, you don't let that little thread of like, am I good enough? Were they right? Is just really, really being your own champion and knowing what you want.
Yeah, I would say the same thing. I would say, be confident in taking space. You bring something to the table, take space.
Yeah. Well, it does get to me, but fortunately or unfortunately, I'm very, very tenacious, and so I think this tenacity
That's better
and
than stubborn. I like that. Tenacious.
yeah. So, I have been described as tenacious by a few people independently in different times. So I guess it's that stubbornness that, yeah, and just do it. Just do it. just go for it. Stop thinking. Just go.
Yep. And I would say also, like I said before, network, build your community. That's very important.
Yeah, those are really, really great points. I think, we converge on a certain themes. What I'm hearing is like, be tenacious, be stubborn, be passionate, really chase what you are passionate about, and don't deter from that, no matter what other people are saying, right? And I actually think I really like what Jasmine, you're saying, that, if you find something that you're really excited about, naturally you're going to go after that. So if you are always worried about, other people, how do they look at me and all that, that means you haven't really found the things that you're really truly passionate about. So, I think I wanted to wrap this up by thank you, all of the wonderful ladies to come in to join me, discuss women's in leadership. I learned a lot from you and from sharing all these fascinating stories. I feel like the time went by too quickly. I wanted to do another session with you already, really deep dive into your science, because those are the really fascinating topics we haven't really touched thoroughly today because we run out of time. But again, thank you so much for your time. I really appreciate it. I'm so proud of you, proud of our women's leaders in genomics.
Thanks for having us, Molly.
Yeah, thank you so much, Molly.
Thank you, everyone. It was
Thank you.